happy to share the work that my lab and David Calderwood's lab did on tousled-like kinases. So tousled-like kinases, or TLKs, were originally identified in plants where mutations in the Arabidopsis ortholog lead to defects in leaf and flower development, providing this disorganized or tousled phenotype. Humans have two TLKs, TLK1 and TLK2, and as you can see by their placement in the human kinome dendrogram, they're quite divergent with respect to their sequence in the catalytic domain. They don't belong to any of these major kinase groups. So with respect to primary structure, all TLKs have a C-terminal serine-threonine kinase domain, and that's preceded by a series of coiled coils that are important for homoligarization. And then there's an N-terminal region that we call the tousled homology, or TH region, that, while predicted to lack structure, is well conserved from plants to animals. In animals, these are broadly expressed in pretty much all tissues. They reside primarily in the nucleus. And TLKs, interestingly, are activated during S phase of the cell division cycle. How this occurs from a mechanistic standpoint is not known. And though a handful of substrates have been identified, the major substrates appear to be the histone chaperones ASF1A and ASF1B. And phosphorylation of these substrates is important in a process called replication-coupled nucleosome assembly. So during DNA replication, the formation of new nucleosomes from both recycled histones and newly synthesized histones requires this extensive network of histone chaperone proteins that serve to prevent histones from aggregating and are important for their timely and ordered deposition onto DNA. So ASF1A and B in particular bind to histone H3, H4 dimers, and they prevent their tetramerization, facilitate their import into the nucleus, and then hand them off to other histone chaperones like the CAF1 complex that assemble them into nucleosomes. And so TLK1 and TLK2, its closest ortholog, they phosphorylate ASF1 in their C-terminal disordered regions, and this promotes the binding of histone H3, H4 to the histone chaperone domain. And in this way, TLKs help meet the histone supply, or help provide a histone supply to meet the demand for during DNA replication when the nucleosome content of the cell is doubled. So we got interested in these kinases because of their connections to human disease. So in the interest of time, I'm just going to talk about their relevance to human cancer. So TLK2 in particular is amplified in a subset of human tumors, most frequently in breast cancer. And it was found that its knockdown impaired cell growth both in culture and also in xenograft models in mice. And interestingly, knocking down TLK2 sensitizes cells to DNA damaging agents, checkpoint kinase inhibitors, and also to PARP inhibitors. And this is sort of interesting because that suggests that TLK2 inhibitors might synergize with current FDA-approved therapies. So at the outset of this project, there was really nothing done in terms of TLK2 inhibitor development. And there's a lot of really basic stuff that we didn't understand about these kinases. And so one thing in particular was that we really knew nothing about the substrate specificity of TLKs. So one of the things that we did early on was to screen a peptide library, and this was using an arrayed positional scanning peptide library format that my lab had developed and adapted to a semi-automated platform. And as was mentioned in the introduction, we're now applying these methods to other dark kinases as well. So TLK2 provided some interesting results. It turns out that it is a sort of unusual, and that is a so-called acidophilic kinase. So it recognizes acidic residues, aspartate and glutamate, at multiple positions upstream of the phosphorylation site, and also has a somewhat unusual preference for polar and aliphatic hydrophobic residues at the position immediately downstream of the phosphorylation site. If you look at the sites that had been mapped in ASF1A and ASF1B, you can see that these residues are overrepresented. So we do see at least a preponderance for acidic residues upstream of the phosphorylation site, and we always see polar or aliphatic residues downstream of the phosphorylation site. So this tells us something about why TLK2 at least targets these sites in ASF1A and ASF1B. But the other thing that this sort of data is useful for is in the design of substrates to be used in drug discovery campaigns. And so we generated a so-called TLK-tide, which is just a consensus peptide where we strung together all of the residues that were most selected at various positions surrounding the phosphorylation site. And we used this as the basis for a so-called kinase glow assay that is suitable for high throughput screening. So far, we've only screened a focused library of known kinase inhibitors. And of course, we identified some weak inhibitors of TLK2, nothing particularly potent, nothing particularly selective. These are compounds that are much more selective for other kinases, but we started efforts to co-crystallize these compounds with the TLK2 catalytic domain, with the idea that we might be able to use this as the basis for structure-guided optimization. Unfortunately, though, we got crystals. We don't have anything that diffracted well. Something that we've been doing now is collaborating with the UNC group to screen the GSK published kinase inhibitor set, hoping that we might find something that would be a better basis for future development. So one of the interesting things that we noted as we were studying TLK2 was that in addition to its phosphorylation site specificity, there also appeared to be non-catalytic interactions that were important for promoting phosphorylation of ASF1A and ASF1B. So that's shown here, where basically if we use full-length ASF1A as or in this case ASF1B as a substrate, it's phosphorylated much more robustly than the C-terminal tail alone that harbors the phosphorylation sites. And I don't have time to go into it, but we were able to map a physical interaction between ASF1A and ASF1B to this tousled homology region at the N-terminus of TLK2. And in fact, we were able to further map this interaction to the first 30 amino acids of the kinase. And then we were able to solve the X-ray crystal structure of the histone chaperone domain of ASF1A in complex with this N-terminal fragment of TLK2. What you can see here is that the peptide adopts a helical conformation when it binds to ASF1A. And what was interesting to us immediately when we saw this structure was that if you compare to the previously reported crystal structure of ASF1A in complex with the H3H4 dimer, you can see that the TLK2 binding site overlaps with a portion of the histone H3 binding site, and they basically bind in the same mode. And so this suggested to us sort of an interesting mechanism for substrate recognition, where TLK2 targets ASF1 and ASFB by mimicking their histone clients. And we were able to confirm that this binding, sorry, what's that?
1 minute.
1 minute. We were able to confirm that the binding interface was important for phosphorylation of ASF1A through targeted mutagenesis. But the other thing, and this is, I'm almost done here. The other thing that was this suggested to us is that we were starting to think about targeting this interface with small molecules. And though, obviously inhibiting protein-protein interactions with small molecules is a difficult enterprise, the type of interaction that we have here, which is a helix globular domain interaction, is similar to that targeted by things like venetoclax and the Nutlin class of p53 MDM2 inhibitors. We also noted that the interaction really centered on this hydrophobic pocket that accommodates one of the a phenylalanine residue in TLK2. So we developed a fluorescence polarization assay using as a tracer this N-terminal TLK2 peptide fluorescently tagged, and we're able to adapt this to a high throughput format where we got nice parameters in a pilot screen. We haven't identified any inhibitors, but what we're right now doing is seeking some support to screen the entire library that we have here at Yale. And of course, we're interested in virtual screening approaches as well. So I'll stop there and just thank the people who did the work. This was, again, a collaboration with David Calderwood's lab. Bertrand Simon did the crystallography, and Jane Liu in my lab did all the kinase assays. And happy to take questions.